Welcome to my YouTube channel. Please like, subscribe, comment, and share. We are doing advanced level biology, the nervous system, and we are looking at the resting potential. So, let us start. Before we look at the resting potential, we need to understand what is called potential difference. Let me just correct that. Change the potential difference. So potential difference is the work done per unit charge to move a test charge between 2 points. So we are saying, let's say we have a point here negatively charged, and you have got a point here positively charged, and you have got maybe a vacuum pump or some wires to transmit the charge. Let's say is the negative charge. So potential difference now is the work done by this electron to move from the negative to the positive. So that is the potential difference. To lighten up very soon when we talk about the resting potential. So potential difference across cell membrane, it's usually negative inside the cell, but with respect to the outside. So in essence, we say our membrane is polarized. So let's say we have this line is our cell membrane, and then this is the outside of the cell membrane, this is the inside. So we are saying that the inside is relatively negative and the outside is relatively positive. Now if we look into our first diagram, we realize that there is now some similarity between these two diagrams. Okay, so let us go into resting potential. So resting potential, now this is the resting potential difference of the cell. So it is maintained by active transport and passive diffusion of the ions. There are certain ions which are actually important for this resting potential to happen. We'll talk about very soon. So it's about the resting potential is about minus 70 millivolts. So as I said, there are certain ions which are very vital for the maintenance of the resting potential. We've got the sodium ions and the potassium ions. So these ions are outside at the fluid outside the axon and in the cytoplasm of our neuron. So the axoplasm is actually the cytoplasm of our neuron. So we have got a high concentration of sodium plus at the outside and low concentrations of potassium ions at the outside. We have high concentration of potassium ions in the inside of the neurons, and we've got low concentration of sodium plus in the inside of the neuron, which is the axoplasm. This will make sense later when will be explaining the resting potential. So chlorine and other ions do not play chlorine and other ions do not play vital roles in the process. Okay, so there is gradient across the membrane. Gradient across the membrane. These gradients are due to some properties. We have electrical properties and chemical properties. When we look at electrical properties, we are looking at charge, and at charge we say uh, opposite charges at opposite charges attract. So that means if the outside of the membrane is positive and the inside is negative, that means the positive charge will tend to move inside the cell, and like charges repel. That means if a an ion is negative, it will most likely not be very supported to move into the cell. We also have concentration. Concentration, this usually moves from high concentration to low concentration. This is on chemical. So we want to look at the maintenance of the resting potential. So resting potential is maintained by active transport of ions against the electrochemical gradient. So we have talked about the two gradients, the electrical and the chemical. So active transport is against the electrochemical gradient, both the gradients. So we have proteins which act is to facilitate this to come to pass. So we have got the sodium and sodium potassium pumps. So let us explain active transport a little more. So active transport is the energy consuming transport of molecules or ions across cell membrane against concentration gradient. So we have got energy in form of ATP. This energy will be used to move substrate against its natural tendency to diffuse in the opposite direction. Usually move in one direction. So it usually moves in one direction. Let's just have a little diagram to explain this. We have the cell membrane, we have the outside, and we have got the inside. So we've got high concentration of sodium ions and got low concentration of sodium ions. Here we've got low concentration of potassium ions and high concentration of potassium ions. So we are saying active transport, it uses energy to move these sodium plants from their areas of low concentration to their areas of high concentration, and it also uses these It's also these pumps, or these sodium pumps with, or the potassium pumps also pumps sodium from its low concentration outside the cell to high concentration inside the cell. So we have said that active transport is the one which maintain the resting potential. At this point, it will not really have come to surface, but let's go on further. Just hang on with me. It will make sense at the end. So we want to look into something. Uh, just an example of how cyanide ions poison the body. So cyanide ions, they inhibit respiration. So if you by this is by blocking the transport chain, electron transport chain. So by blocking that, they prevent the manufacturing of ATP. And we said ATP is used by active transport. So that means on the membrane there will no longer be this active transport which we were talking about. So ion composition gradually changes. Uh, Ion composition gradually changes. Uh, then equilibrium happens with the plasma. That means the environment, the outside in the inside will become equal. That means there won't be our resting potential anymore. And eventually you will see by osmosis water will start to move in because the sodium is moved in and the cell will disrupt. Okay, let's look more into the sodium pumps in the potassium pump. So the sodium pumps, this actively pumps sodium ions out of the cell because that's the direction of the low concentration to high concentration. And then the sodium pump, they actively move potassium ions into the cell. Okay, so they are what are called sodium potassium pumps. This is a combination of both the sodium pump and the potassium pump, and they are usually a common feature in all animals. So what's a pump? A pump is a carrier protein. So this carrier protein inside is accept ATP and sodium, and outside is accept potassium. Whereas outside is accept potassium. But now it's not. There is a ratio on the ions. This is the sodium ion, the potassium ion. There is a transfer ratio which occurs. It's 3:2. Okay, let's just draw our diagram, our previous diagram, so that they will better understand. So we say this is our membrane, cell membrane, and this is the outside, and this is the inside. And you say we've got high concentration of sodium plus, and we've got low concentration of sodium plus, and you say we've got high concentration of potassium plus and low concentration of potassium plus here. So we said a, a sodium pump, a sodium pump on its own, it pumps sodium from low concentration to high concentration. This is outside, and then the sodium, the potassium pumps the sodium ions into the cell. So now we have what is called the sodium potassium pump, which you have just been introducing. So we say the sodium potassium pump, it accepts ATP and sodium ions inside the cell, and then produces it brings them out. And you say their ratio is 3:1. That means there are going to be 3 sodiums out. At every moment there will be 2 sodium in. So if you look at it, there's already a difference, because 3 are going out and 2 are going in. That means the inside of the cell is going to be negative. So the active transport is one of the the means of uh, maintaining the resting potential. As we have seen, this is already making the inside of the cell negative and the outside more positive. That's producing our 2 points we were talking about potential difference. Okay, so let's move on further. So potential difference is built up across the membrane. So as you say, potential difference, then as we have said, we have talked about electrical gradient and chemical gradient. So this is an electrical gradient which we have been creating in the intrinsic entry of anions. These are negative charged molecules or ions. So both concentration and charge are important in deciding the direction in which ions cross membrane. Okay, so let's look into the importance of the pump. So the pump is essential in controlling osmotic balance of animal cells, and also, yeah, looking into the dangers of inhibiting the the pump. The cell swells and it bursts. uh, this is because of the buildup of the sodium ions, resulting in excess water entering in the cell by osmosis. And then if we look into bacteria, fungi and plants, they have got no need of the pump because they have got cell walls. Then we are seeing that the sodium ions, when they enter into the cell, that's when the osmosis start, the water start to move in by osmosis. But when the sodium ions are in high concentration, that won't be happening. This is because sodium ions are used in potassium ions. Sorry, potassium ions are used in photosynthesis, protein synthesis, and glycolysis. So active movement of ions active movement of ions uh, opposite active movement of ions is opposed by passive diffusion. This is movement of ions down their concentration gradient through ion channel proteins. Now these are ion channels, not sodium potassium pumps. They are not pumps now. So there is a difference in the rate of diffusion. The rate of diffusion is determined by the permeability of the axon membrane to the ions. So the axon membrane is more permeable to potassium ions. It's 20 times than that of the sodium. We'll shortly draw the diagram so that we can fully understand what this will implicate. So sodium plus ions in sodium plus loss is greater than sodium plus gain. So we are seeing that because of the permeability ratio, the sodium plus which moves in from high concentration to low concentration are actually there is more loss of sodium plus than the gain of the sodium because of the ratio which is at which this is happening. So there will be net loss of sodium plus, hence the negative charge of the axon in the membrane. So the value of the resting potential is likely determined by sodium plus electrochemical gradients. Okay, so now we have all the details which we need. We now want to give a simple explanation of this whole resting potential. So we've got the cell membrane, and we say we've got the outside. This is the surrounding environment or fluids. We've got the inside. This is the cytoplasm. So we first said we have concentration. We have important ions which takes place in this resting potential. So we have sodium ions and potassium ions, and we say this you have different concentrations inside and outside. And we said outside we have got high concentration of sodium ions, and in the inside we have got low concentration of sodium ions. And also we have potassium ions. We have said they have got high concentration in the inside. These are potassium ions. And then we have got low concentration of potassium ions at the outside. Now we have what we call the okay. Before we go to the let's go to the sodium potassium pump. We said we have got what we call sodium potassium pumps. So sodium potassium pump, they accept sodium ions and sodium ions plus ATP. These go outside, and we said this you have got these are three which go outside. They go outside in the ratio is 3:2. And we said this is active transport because you are moving from area of low concentration to high concentration. Again, for potassium ions, we are moving from high concentration to low concentration. This is, as we have said, this at every two sodium ions which go out. There will be two potassium ions which go in. So these are charged ions, which means, let me just write this potassium ion. This means the inside of the cell will become slightly negative. This will become slightly negative because we have removed three ions in the cell and bring out in two ions with the same charge. This is the same charge, so it's more like we have just removed three positive charges and then retained the two positive charge. So that means one positive charge is remaining at the outside. So that means this will become, the outside will become slightly positive, but this positivity is very slight, so it needs to be helped by what we call transport chains. We said transport chains. Now we've got sodium ions which are moving in from high concentration to low concentration. This is a passive diffusion. So and we also have at this side, we also have the same channels moving from high concentration to low concentration. Now, if this was happening at the same rate, we could say the charges only be determined by this, by the active transfer. But this is not so. We realize that this one is 20 times the sodium plus the potassium plus ions are 20 times more diffusing. So we can say 20 potassium out. Here we have got one sodium in. So you can see the difference. In every 20 sodium ions which goes out of the cell, there's only one sodium ion which goes inside the cell. So the difference in charge is so high that the negativity of inside of the cell become very high, and the positivity is outside become and the outside becomes more positive because more ions are being brought in outside the membrane, and more ions are being brought in, less ions are being brought inside the membrane. If you look at it, uh just combining, you can see that there are three ions outside plus the 20 plus the 20 going out by diffusion, and here we have got two going in by active transport and just one going in by active by diffusion. This is the first one is by active transport. So you realize that we have about 23 going outside and 3 and only 3 going in. So the difference, you see, these are all positive charge, positive charge. You realize that the outside will become more positive and the inside will become more negative. So this is our resting potential. So thank you for watching. Please follow and subscribe, comment, share. Yeah, thank you so much.